Welcome to our Environmental DNA Workshop version 2024. This is an introductory lecture, a short one, hopefully that will last about 30 minutes, where we're going to talk about some of the definitions, origins, and fates of eDNA. So let's begin with defining DNA and speaking about where eDNA actually comes from. Environmental DNA in the broadest sense is simply DNA that derives from organelles like mitochondria, chloroplasts, or the nucleus that is found in any environmental matrix. And that can include water, so fresh water, brackish water, or marine, from soil, from air, it can come from ice cores, it can be found in permafrost, it can also be found on the surfaces of leaves. It can be found in other environmental surfaces, biofilms, and also in feces. Although we will be emphasizing eDNA methods, I want to draw your attention to eRNA methods, which can be used in programs like COVID-19 and other pathogen surveillance programs of wastewater. But RNA also has some signatures and some characteristics that make it particularly useful for looking at short time frames or looking at physiological activity in nature. And we'll make allusions to this throughout the lecture. So let's talk briefly about eDNA sources. eDNA, as you will undoubtedly know, can come from decomposing carcasses. So when an animal dies and starts to degrade, ultimately the cells will lyse and DNA will be released into the environment. It can come from living organisms through shed epidermal or epithelial cells. For example, fish or amphibians have a mucus layer, and this often will be released into aquatic environments. Reptiles shed their skin as they grow, and reptile skin is a rich source of DNA. Other body secretions can deposit DNA into the environment, including feces and urine. And of course, when there are mating events, and particularly when we're talking about mass mating events as occurs in anadromous fishes or on coral reefs with coral species, this kind of mass event puts a lot of DNA into the environment. When we sample environmental DNA from either soil or water particularly, but also from feces and other media, we also include living organisms. So we'll take whole living bacteria, viruses, phytoplankton, microfungi, invertebrates, algae, all of those different things will be incorporated. And so when we extract DNA, there are two different components, really, that derives from living organisms and that which derives from the remnants of living organisms and from DNA. We can, and you'll see this in the literature, split DNA into that which is extracellular DNA, so DNA that is free and floating in whatever medium you're speaking of, and also intracellular, or this is versus intracellular DNA. And this is DNA that is still within organelles, still within the nucleus, or is associated with membranes and other parts of cells, whether it be a living organism or an organism that is recently deceased. To understand the power of eDNA and the limitations thereof, we have to understand what happens to eDNA once it's released into the environment. And that's what we're going to do here. So a lot of the literature that we'll talk about comes from aquatic environmental DNA, and particularly freshwater eDNA. And this is simply because that is where the field really started. So we can talk about the half-life of extracellular eDNA as opposed to that which is associated with cellular debris. And it can last just a few days in particular environments, and we'll speak about this in just a minute, up to millennia if the DNA becomes absorbed to particles that are deposited in sediments. In this paper, which was a very nice summary that has been cited hundreds of times, put out in 2011, really near the beginning of environmental DNA as a means for monitoring environments, they reported that DNA typically in freshwater lasts only between a week up to 3 weeks. What this means is that if you detect the signature of a species in an aquatic environment, typically that means that the species is present or has recently been present. So it's a nice temporal snapshot of, of the presence of a species. eDNA persistence depends on multiple things. It depends on the DNA attributes. So we'll return to this, but if it's a circular molecule like a plasmid, or if it's a circular molecule, as we find in the genomes of plastids, then this tends to resist degradation more quickly. The length of DNA is important, so longer pieces will degrade into smaller or shorter lengths of DNA, or RNA for that matter. And whether it's associated with cellular or organelle membranes or proteins like histones will shape the fate of environmental DNA. We also can speak of abiotic factors that might accelerate or protect eDNA and cause a more durable lifespan. So for example, light, oxygen, whether it's in an anoxic environment or oxygenated, pH, salinity, substrate abundance and composition, whether if we're talking about an aquatic environment, whether we're talking about a turbid environment, and the temperature. These are all factors that can impinge on persistence of DNA. And then finally, we must consider biotic. So if there's a lot of microbial or algal activity in a particular water body or particular soils, or if there's a lot of extracellular enzymes, this can certainly shape the fate of environmental DNA. I've given you a few references here, 4 references on this slide, and we'll try to make some of these papers available to you so you can read them if you wish yourself. But ultimately, it's a rather complicated process when we speak about understanding in some deep way the persistence of environmental DNA. So this is let me just move this up a little bit. I don't need to be front and center. This is a very nice summary of the ecology of environmental DNA that was included in this paper by Barnes and Turner in 2016. And it really summarizes some of the interesting aspects that we have to contemplate. And it does mirror the complexity of understanding all facets of environmental DNA. So I spoke briefly about the origin, where eDNA might come from, and this is a summary in the upper left-hand panel. One of the things I'd like to draw your attention to is the possibility of secondary eDNA signals. So if we're talking about a predator, perhaps eating a fish in one water body, like an osprey, for example, but defecating in another water body, you might see DNA released from that fish prey species into a water body where that species doesn't exist. That's a form of secondary signal. I've already spoken to the state of eDNA. It can be intramembranous, extramembranous, so but associated with cellular membranes. It can be particulate, and it can be free or dissolved. The fate of eDNA, I've alluded to, so we can get a whole variety of different processes in nature that will ultimately degrade DNA from longer stretches of DNA sequence contiguous down to shorter. One interesting consequence or fate of eDNA that you may or may not be aware of is horizontal gene transfer. So bacterial species may take in some DNA from the environment. There's a very interesting phylum of aquatic organisms called rotifers. And within rotifers, there's a group called the Deloid rotifers. And they're notorious for taking in bits of environmental DNA to repair environmental or to repair DNA damage. And so there are different genes from different organisms. And it's a very messy evolutionary tree. If you read about them, it's a very interesting phenomenon. And then we must consider transport. So eDNA doesn't just sit in a medium, it may diffuse to other parts of the medium or the region that you're sampling. There are a whole bunch of other things that we must consider. Biological transport. We have to think about anthropogenic transport. So for example, bait fish might be transported among different water bodies. We might see foreign species introduced into water bodies because of human activities, and so on. So this is something that we really have to think about when we're trying to interpret eDNA signal, and we'll return to this multiple times. So if you put this all together, here is a very nice summary of the technical aspects that one must consider and the challenges therein. And this class, this course, this short workshop is going to go through some of these in some detail so that you'll become aware of these. And this really enhances your ability to interpret environmental DNA literature, but also to design your own studies. So as I said, understanding the persistence of environmental DNA is complex. This is just another breakdown, a more recent paper, and it talks about the multiple states of environmental DNA and how environmental DNA might be broken down from long contiguous stretches into its constituent parts. So first we can contemplate intracellular or intra-organellar DNA, so within a mitochondrion or within a chloroplast, and we can see degradation by intracellular hydrolysis. So this is enzymatic digestion that is facilitated by the presence of water. If these cells are lysed, you might end up with dissolved DNA that can be absorbed to particles within the environment. And interestingly, from these absorbed particles, you might actually get re-release of DNA back into the medium. And that is a source of environmental DNA that we might wish to contemplate when trying to interpret the DNA signatures of particular species. Dissolved DNA can undergo hydrolysis outside of the cell or chemical modification and ultimately be consumed by microbes. The most important thing, I suppose, with respect to adsorbed particles and sediments is that these might accumulate over decades and even centuries and millennia. So if one takes a core from sediment within an aquatic environment, you might be able to recover the signature of DNA over time. And indeed, in our laboratory, particularly Stafford, who you'll hear from later on in this workshop, that is exactly what we're trying to do. As we will come to understand, most of the markers that we use in the techniques that are deployed in eDNA studies focus on organellar genomes or plastid genomes. And so we want to ask the question why this is. So first, let's just make the case or come to understand or refresh your memories that both mitochondria and chloroplasts have their own genome with a small but stable number, or typically stable number, of genes. And so that means that we have DNA sequences from the nuclear genome and we have DNA sequence from the mitochondrial genome. And in animals, it's mitochondrial only, but in plants, you have chloroplast and mitochondrial DNA. I wanted to draw your attention to this. This is a recent study. This is a new organelle that has been discovered in some marine algae species called a nitroplast, and it too actually has genomic machinery. This is in the process of completion of endosymbiosis, which is the way that we got mitochondria and chloroplasts, so the engulfment of one cell by another, and then the gradual over evolutionary time coordination of metabolic processes. And that is what seems to be happening here. So perhaps in the future, we might focus on genes from the nitroplast to employ in eDNA barcoding. So back to the question at hand, why do we use mitochondrial or chloroplast genes in DNA barcoding? There are a couple of simple reasons for that. Typically, there are many more copies of organellar than nuclear genomes. And there are a couple of reasons for this. Typically, we see in animals, particularly 10 to even hundreds of mitochondria per cell. What that means is there are tens to hundreds of copies of the mitochondrial genome for every single copy of the nuclear genome. And even more spectacular still here, within organelles, you can have multiple copies of an organellar genome. So it's not just one mitochondrial genome within a mitochondrion, it might be tens of them. This varies, of course, according to tissue type and function, stage in the cell cycle, age of the individual. But the upshot of all of this is when you get lysis of an organism or cells of an organism, you will end up with a lot more organellar DNA in the medium at hand than you will nuclear DNA. And this greatly improves your probability of detection of the presence of that species. So that is why we use this. There are a couple of other things that we must consider here. One is that mitochondrial and chloroplast genomes, plastid genomes, typically are circular. And circular DNA tends to resist digestion or degradation better than linear. And so potentially these genomes, when initially released, are more persistent. And then finally, there are large amounts of data in databases like GenBank, and GenBank is an NCBI repository. NCBI stands for National Center for Biotechnology Information. And then we also have the BOLD system. So this is the Barcode of Life Data Systems. And both of these are repositories for large amounts of DNA. But when the fields of phylogeography and phylogenetics were in their infancy, for the same reasons that I just indicated previously, mitochondrial DNA particularly was more accessible. So a lot of the initial studies in phylogeography and phylogenetics used mitochondrial genes like 12S, 16S, cytochrome B, and cytochrome oxidase. And these are the genes that really have become the workhorses of DNA barcoding initiatives. So large repositories of sequence data that really is a historically contingent event, if you will, that we started with these large repositories. We can access them. We can search an unknown sequence against these large repositories. So that's another reason we tend to focus on these mitochondrial or chloroplast genes. So let's speak a little bit about the methods that we're going to talk about in this workshop. We're going to talk a little bit about traditional PCR, but we're really going to assume that you have some knowledge of that. And then we're going to talk about loop-mediated isothermal amplification, or LAMP for short, which is a relatively new technology that doesn't require a lot of power, is incredibly sensitive for detecting single species. We're going to talk about quantitative PCR, or sometimes called quantitative real-time PCR. The problem with RT-PCR as an acronym is there's also reverse transcription or reverse transcriptase PCR, so it gets a little bit confusing. We're going to talk about digital PCR, and we're going to talk about DNA metabarcoding. And we're going to work through the theory of this and work through some examples. We'll also allude to metagenomics, and it's in the manual, but we're not going to speak about that in any detail in this workshop, because this is an awful lot to cover in one single 1 week workshop. We're going to return to the benefits of environmental DNA and the limitations of environmental DNA a little bit later. Delaney will speak to this in some detail. But just very briefly, eDNA has been favored because it's non-invasive. In other words, you simply have to sample water or soil or air or some other medium without ever having to touch the organism. It means you don't need permits for animal care or those kinds of things, because you're never going to come in contact with them, with the species that you're focusing on. It can be useful for rare, cryptic, or elusive species that might leave their DNA signature but might not be obvious in at a particular site at a particular time. Breeding may already have occurred. They might be crepuscular or nocturnal. They might not vocalize, all kinds of reasons. The other nice thing about eDNA is you can contemporaneously sample across a huge array of taxa, and you can really build up a geographically intensive profile of either biotic assemblages or single species. There are lots of limitations of eDNA that we have to be cognizant of. It is not a panacea for all challenges in surveying biological diversity. It is indirect, so the signature of the presence of a species doesn't mean it's necessarily present right at that particular site. The DNA may have come in from somewhere else. And sort of a corollary to that, or an extension of that, is just because you detect eDNA doesn't confirm the presence of the focal species without direct confirmatory evidence. eDNA, as we will see, and particularly some methods over others, can be subject to contamination, and this can lead to false positives. But in some environments and in some samples, you also can get striking inhibition and end up with false negatives. And so one has to be aware of these possibilities when interpreting DNA signatures. There are lots of different applications of environmental DNA, and we'll touch upon lots of these, and hopefully you'll have lots of questions for us, and we can explore these in some greater detail. eDNA can be used as an early warning for invasive species when just a few individuals might be present in a particular region or environment. And so this can raise the alarm and sort of drive some mitigation measures very early on. It can be used for tracking species of conservation concern that often, by definition, are quite rare. It can be used for the surveillance of pathogens, not just COVID-19, although that's the most obvious case. But as an example, we're using it to track chytrid fungus presence at the Queen's University Biological Station. It can be used in algal bloom monitoring, and we've done this ourselves in our lab. It can be used to assess species distributions and range limits, and this is particularly relevant in areas which don't have a lot of human habitation, where we may wish to understand northern range limits and model what might happen in the future. And in this domain, niche modeling using eDNA might be relevant. It can be used as a metric of ecosystem health, and here we might want to get at the totality of biological diversity in a particular system. And finally, just more generally, it can be used for quantification of different aspects of biological diversity or for assessing species richness to address a whole host of questions that I'm sure you all will have at the tips of your febrile brains. I did want to indicate that eDNA has really risen in popularity, and there has been an explosion, as you will see, of literature and applications. So eDNA has been or is being incorporated into all kinds of different biomonitoring. So here's just one example. It's a local, and we're sort of indirectly involved in this. So this is a collaboration between the Invasive Species Center and the Federation of Ontario Cottagers Association. And the intent here was to track zebra and quagga mussels and their spread. This is called iSample Ontario. It depends on community or citizen science, which is to say that cottagers associations will sample the water using a kit, send the water to the Invasive Species Center, who will then send it to a laboratory for processing, and then the data and interpretation therein is returned to each cottage association. This is probably the most famous example of application of eDNA. You will probably know that Asian carp, 4 species, not just one species, like this big head carp, have been introduced into riverine systems in the United States. And there was some fear that they might breach some barriers and get into the Great Lakes. And that's what these maps show. So here is Lake Michigan, the southern terminus, and there's some concern that we might get Asian carp coming in from further south here. And so there has been developed some eDNA tools, quantitative PCR tools, to try to detect the presence of Asian carp and to try to monitor annually and test for the presence of Asian carp. And so this is an interesting and early example of eDNA with invasive species. There are lots of other community-based projects in the United States, in Canada, and around the world. This is out of the University of Guelph and is a reasonably new community-based project which involves a collection of bulk DNA samples from rivers across Canada. The name of the consortium is Stream DNA, Stream standing for sequencing the rivers for environmental assessment and monitoring. And partners include Illumina, which is this large company that builds sequencers and provides sequencing kits, and we use them a lot in our lab. And it also involves a partnership with ECCC, so Environment and Climate Change Canada. And there are lots of different commercial services that are coming online. So a lot of graduates of different programs seeing the promise of environmental DNA are creating these companies. So this is one I found just with a very simple search out of St. John's, Newfoundland in Canada. It's called eDNA Tech. And they do environmental genomics, but they do a lot of, or propose to do a lot of environmental DNA work. So that's kind of exciting. So just to wrap up, I wanted to present this, and we will provide PDFs of these lectures, so you'll have all of this at your fingertips. But this is a summary of eDNA metabarcoding for biomonitoring. And it's a little bit incomplete. It's mostly focused on terrestrial and marine, so it doesn't include air. It doesn't include some other sources of environmental DNA, but it does give you a nice overview of the kinds of things that one can look for. So in a terrestrial environment, you could perhaps look for microbial diversity in soils, fungi, parasitoids, arthropods, not anthropods, a whole bunch of things. You can do that in sediment as well. Here is just a summary of some of the other kinds of organisms that you might use water for marine systems to search for, freshwater systems, a whole host of species. Just as an aside, one of the examples we'll talk about or return to in our laboratory is an effort that we undertook to try to essentially come up with a barcoding schema that would allow us to survey all of these different groups simultaneously. Using DNA metabarcoding, and we'll talk about that in some detail. But this gives you a broad idea of the power of eDNA, and particularly in this instance, the power of eDNA metabarcoding. And we look forward to returning to this at various junctures and discussing this with you. And we really look forward to spending time with you in this workshop. So thanks for your time. We'll see you in person shortly. And Delaney, Bogen, Stafford, Alan, the staff of the Queen's University Biological Station, and I look forward to spending some time with you. Thank you.